I am Professor Shyamapada Mandal, Gour Banga University, India. Good evening and good afternoon also to all, and especially the Drug Discovery Scholars Conference Organizing Committee. My today's presentation is computational methods in the development of small molecule inhibitors against SARS-CoV-2 multi-drug approaches. So now actually the title of this paper is the plant-based active ingredients of some homeopathic medicine in the development of therapeutic effective against snake bite envenomation. This is an in silico study. So snake, this one snake we know the in India, Russell viper, Vipera russelli and Pluchella. From this snake, a venom is uh, extracted, which is known as phospholipase A2. This phospholipase A2 enzyme has been utilized as a drug target because this enzyme is responsible for the breakdown or hydrolysis of membrane phospholipid. And due to the membrane phospholipid hydrolysis by phospholipase A2, arachidonic acid is formed that is responsible for the formation or elaboration of two pro-inflammatory molecules which are known as prostaglandin and leukotrienes, and these are inflammation, these cause inflammation. So after snake biting due to phospholipase A2 activity, inflammation occurs and it is actually a neurotoxin or myotoxin. That means it causes effect on nervous system or as well as muscle, causing inflammation, and death may occur if not uh, actually it is treated or not antivenom substance is applied. So under this background, we know that in India and in other parts of Asia, in the world, this viper is present and envenomation occurs due to snake biting in different ways, particularly for the persons who are working in the field, and death also occurs. So this is an urgent need to prepare the therapeutic agents for the treatment or as an antidote. And we know cedron, a homeopathic drug, which is a plant-derived substance, is an known is a known antidote that is applied after this envenomation. That means the snake viper Lucella. And molecular docking we have performed with this target that I have already told, phospholipase A2 enzyme, and ligand preparation, protein preparation, molecular docking, 3D interpretation and 2D interpretation of DOCS complex, that is phospholipase A2 with phytochemicals and cedron as a reference drug. And after that, drug likeness and admit analysis of the ligands were performed through Lipinski's RO5, rule of 5, bioavailability radar chart analysis and admit property analysis. And the softwares utilized here are UCSF Chimera version 1.15. This is the website from where we can retrieve the software for analysis. PyMol was utilized, Open Babel version 3.1.1 utilized for file conversion from one type to another. BioVia Discovery Studio Visualizer, this version was utilized for the interpretation or analysis of docked complex. That means the phospholipase A when formed the complex with phytochemicals. And PKCSM and SwissADME, these are the two web servers which are freely available to interpret the pharmacological properties of the phytochemicals. RCSB PDB, from this website we have retrieved the 3D structure of protein phospholipase A, which is the target in our study, and this is actually the snake venom from a Russell viper which are found in India and other parts of the world in Asia. PubChem, this is responsible for a drug bank, and from where different small molecules, bioactive molecules, can be retrieved in their 2D or 3D forms or their smiles for the analysis of drug likeness properties and other properties, other pharmacological logical properties. And these are the 6 ligands which are used against phospholipase A enzyme. That means the Russell viper's venom. First one is andrographis. This andrographis extract, this is the depiction formula or the configuration of the phytochemical, bioactive phytochemical andrographis extract. From PubChem, with this CID, we can retrieve similarly alkaloid C from Gelsemium semiparvum with PubChem CID. It can be retrieved, and melandrine plicatic acid from Thuja plicata extract, and hypericin from hypericum. So these are the 5 phytochemicals that can be extracted from 5 different plants and utilized as active ingredients of homeopathic medicine, which are used in the treatment against different types of infections such as virus, bacteria, or against inflammation, and also useful for the treatment of depression as well as other neuronal disease. And Cedron, this is the reference drug which is actually are in use against phospholipase A enzyme envenomation due to snake bite, and this is an antidote which are actually in use in current stage. And now we have seen that this is the protein structure phospholipase A, and from protein data bank RCSB with this ID 2B17 it is retrieved. It is actually contain a ligand which is the diclofenac. That means this protein phospholipase A2 already formed a complex with diclofenac, which is available in this website in protein data bank, and that was retrieved by us. This is the 3D structure, and after that we have prepared this protein with the removal of this ligand. These are the water molecules and ions, all were removed, and this is the prepared protein for docking, molecular docking. So you will study for its inhibition by the 6 phytochemicals that we have already shown in the earlier slide through molecular docking in silico. And this is Ramachandra plot analysis of the protein phospholipase A2 snake venom. We have analyzed this for its structural authentication, and we have seen that 90 amino acids, that means near about 90% amino acids are located within the most favored regions, that means the red region, and in deep yellow region 11, that this is the additional allowed regions, and there are no amino acid residues located in generously allowed region and disallowed regions. That means in the light yellow region or in white regions of the Ramachandran plot. So this structure, the 3D structure PLA2 snake venom is very useful to be utilized as a receptor or target for the drug development. So we have utilized 6 phytochemicals against this protein through molecular docking. And before docking, we have prepared the grid, and this is the dimension of the grid in Armstrong for the phospholipase A protein, and center, this is the center for phospholipase A protein. And UCSF chimera, in which Autodoc vena is inbuilt, was utilized for molecular docking of the phytochemicals to the PLA protein, that means the snake venom. So this is after docking, we are seeing that 6 phytochemicals are located. This is the green in color. This is the protein, that means the venom enzyme structure, and 6 phytochemicals are located within different active sites of the protein. Andrographis, this is cyan in color. This is located here with magenta color cedron. Cedron is actually the reference antidote that are being utilized in current time for the treatment of persons who are bitten by or envenomated by this snake phospholipase A2 venom. And yellow color, this is gelsemium, hypericin, light pink color, this one, this is the large structure, and melandrine, this is gray in color, and blue in color is the thuja plicatic extract. So all these were docked with the phospholipase A2 enzyme, and there were different types of interaction that we will interpret in the later slides. And this is actually the diclofenac This is in red in color. Already this is available with docked with the protein phospholipase A2 that we have retrieved from protein data bank. And so we are seeing that this diclofenac was located within the active site where yellow color gelsemium and gray color melandrin are located. So these are active to inhibit the structure along with the other phytochemicals that are docked against phospholipase A2 enzyme. And this is andrographis that form due to the interaction with different amino acids, and actually a number of van der Waals interaction are found, and only two hydrogen bonds which are green in color, aspartic acid and cysteine, are formed in this interaction. And due to this interaction, binding energy was achieved, which was minus 7.4 kilocalorie per mole. So the affinity of andrographase 2 phospholipase A2 protein is good. That means it can be utilized as an inhibitor of the enzyme phospholipase A2, which is the venom, and that can be elaborated due to snakebite, Russell viper. And these are the bond interaction between phospholipase A2 and andrographis. This is gelsemium, the second phytochemical and the active homeopathic ingredient, and this also interacted well with the phospholipase A2 enzyme. And in this interaction, we are seeing that different kinds of bonds, the hydrophobic bonds, which are in color, and in green color, the hydrogen bond are formed. And due to such kind of interaction, this is 3D interaction and this is 2D interaction, this produced binding energy minus 7.4 kilocalorie, just as andrographis. And so the affinity of both the phytochemicals andrographis and gelsemium are equal. And here are different types of bonds, 14 bonds, but we have seen in andrographis only 2 hydrogen bonds are produced, but here 6 hydrogen bonds are available in this interaction. And this is melandrin, the third active homeopathic ingredient, and the residues, amino acid residues of this enzyme, the venom, are shown in 3D form, and this is the structure of melandrin, the homeopathic drug. And this is 2D interaction. Here we are seeing that along with hydrogen bond through glycine and histidine, other hydrophobic bonds which are presented in different colors are formed. And here the binding energy in minus 7.6 kilocalorie per mole. So its affinity is higher compared to the andrographis and the second phytochemical. And this is bond interaction. Here also 14 different types of bonds are formed. And hypericin, this is a very active phytochemical, active homeopathic ingredient that displayed binding energy very low, minus 9.1 kilocalorie. That means its affinity towards the venom is very high due to its very low energy compared to the other 3 phytochemicals that are represented. And here we are seeing that 7 7 hydrogen bonds which are presented in green color are formed, and 4 hydrophobic bonds are formed. So due to such kind of interaction, binding energy was minus 9.1 in the phospholipase A2 and so is a very good inhibitor, but the binding energy was comparatively high, which was minus 6.3 kilocalorie per mole. That means this is greater than minus 6.5. We know that when the value is less than or equal to minus 6.5 kilocalorie per ml, the binding energy, then the phytochemical is very active, but it is moderately active against such venom. And the different types of bond interaction in 3D and 2D complex are shown. And also this is a very good inhibitor. And this is the 18 number of bonds which are found in this interaction between phospholipase A2 enzyme and thujaplicatic extract, the homeopathic ingredient. This is Cedron, the reference antidote, which is used against envenomation of phospholipase A2 venom released by Russell Viper due to its biting, and its binding energy is also good, which is lower than minus 6.5 kilocalorie per mole. So this is very active also, and the reference drug and the value is minus 6.9, and the bond interaction are here. The van der Waals bonds are formed, which are 7 in number, and only 2 hydrophobic bonds are available in this complex after docking, but no hydrogen bond are available in this interaction. And these are the 4 number of bonds are shown. And after that, we have subject the phytochemicals, the homeopathic drugs through different testing, Lipinski's rule of 5, bioactivity radar chart analysis, and admit analysis for pharmacokinetic properties of the homeopathic ingredients. And this is admit by this test, we can predict the absorption, distribution, metabolism, excretion, and toxicity of the phytochemicals to be utilized as drugs against any disease or infection or other kind of inflammatory substance, such as this venom phospholipase A2. And the properties were determined or predicted into openly available web server, which is Swiss ADME and PK CSM tools. And now we are presenting the Cedron. This is referent drugs. All the 6 criteria, lipophilicity, size, polarity, solubility, flexibility, are located within the pink region. We know that when the values are located within the pink region, this is producing bioavailability score less than greater than 0.5, and here we are seeing that the value is 0.55. So this is a very good orally available drug, and we are seeing that Lipinski rule for drug likeness property, the molecular weight is less than 500. This is 220 gram per mole, and the number of donor hydrogen bond is 0, less than 5, and acceptor bond is 1, less than 10. And so we are seeing that this obeyed the Lipinski rule of 5, and the gastrointestinal absorption for this Cedron was very high, but there was no blood-brain barrier permeation. And this is the admit properties are represented here, and EMS toxicity. No EMS toxicity was found for Cedron, that is utilized as a antidote against this envenomation, phospholipase A2. And here, hepatotoxicity is not available, but skin sensitization is found, but its oral rate acute toxicity LD50 and oral rate chronic toxicity LOAEL values are high. So it is tolerated in human body if it is applied as a drug, and the patient may be cured if it is administered in proper time. And this is the another phytocomponent which is utilized as a homeopathic drug, and the andrographis extract. All the values within the chart are located in pink region. So the bioavailability score is 0.55, and it also obeyed the Lipinski rule of 5. That means it is bearing good drug likeness properties, so it can be utilized as a drug. And the admit properties are presented here, and we are seeing that this is negative to EMS toxicity, and there was no hepatotoxicity or skin sensitization, and a very good component to be utilized as a drug in future. And the values for gelsemium are also located within the biodata charts pink region, and the value of bioavailability score is 0.55, and it also obeyed the Lipinski rule of 5. So it is also a very good drug component, and also inhibited the protein very effectively with low binding energy. And admit properties are represented here. EMS toxicity negative, hepatotoxicity slightly present, but its high value of oral rat toxicity is high, 2.708, and hepatotoxicity. Still, due to its hepatotoxicity, it can be utilized because of its good effectiveness against the protein, and further analysis can be done. And this is melandrin, only one features which is in saturation. That means the fraction CSP3 value which is less than 0.25 and less than 1 and greater than 0.25. This is beyond the range of this normal value. So the insaturation value is beyond the pink region. Still, its bioavailability score is 0.55. That means it is orally available drug, and its gastrointestinal absorption is high, and it also obeyed the Lipinski's rule of 5 with one violation for also there is no violation, so it is a very good drug like component, and its admit property is represented here. No M's toxicity was found, no hepatotoxicity, no skin sensitization was found. And this is the hypericin. And the last one, it's two component, insaturation and the insolubility. That means the log ESL value, which is not within the permeable region, permeable limit, and saturation also. So its two components are outside the pink region. Still, it bears the bioavailability score 0.55, which is good for a orally available drug, and it disobeyed one feature, which is bond donor, which is 5. So one violation may be found, but we consider for drug likeness property one violation, and more than two or more violation may reject the phytochemical to be prepared as a drug. So this hypericin, which showed very low energy, low binding energy, minus 9.1, is very good drug to inhibit the venom phospholipase A2, and also showed the pharmacological properties within the acceptable limit. So this is a very good drug, and its admit property are presented here. We are seeing that it has no M's toxicity, no hepatotoxicity, and no skin sensitization. The very good properties for a future drug. And this is the thujaplicatic acid, and actually the hydrogen bond acceptor number is 10. It may be with one violation, and its bioavailability score is 0.11. So it is a inhibitory substance, inhibitory phytochemical, but its bioavailable score is low, but and gastrointestinal absorption low. So it is not a good component, but can be utilized if it is further modified through different types of chemical reaction for its better activity as well as better drug likeness property. And these are the admit profile, and we are seeing that M's toxicity is negative, hepatotoxicity is negative, skin sensitization is negative. So this can be utilized as a future drug, though it has lower bioavailability score value, or it disobeyed Lipinski rule with one violation. One violation is accepted. And this is the finally the TPSA, topological polar surface area, versus W log P. This is W log P, and this is TPSA. This is actually the boiled egg plot. One is white region of the boiled egg system, and this is yellow part of the boiled egg system. We are seeing that cedron, andrographis, melandrin, and gelsemium are located within the white part. So when these phytochemicals are located in white part, this is good for gastrointestinal absorption. But among four, only cedron is located in yellow region, and when it is found in yellow region of the boiled egg plot, then it can be concluded that it has blood brain permeation, so it can be reached in the brain and can be available for the neutralization of the venom when it is found in the nervous system, because it is a neurotoxin and also as a myotoxin. When it is myotoxin, then andrographis and melandrin or gelsemium can be utilized, because these are actively absorbed in the intestinal wall. But hypericin and thujaplicatic extract, these are also very good immunity against phospholipase A2, but these are neither cross BBA. This is good for a good oral drug, but this their absorption in the intestine is low, with low rate. Still, this can be utilized because of their low toxicity. So finally, the highlights: molecular docking authenticated the inhibition of phospholipase A2 protein, which is a venom of Russell viper associated with snake envenomation by plant-based homeopathic active ingredients. These are andrographis, gelsemium, hypericin, melandrin, thujaplicatic extract, and cedron. This is reference drug, and these showed that has already been explored. Their binding energy was respectively minus 7.4, minus 7.4, minus 9.1, minus 7.6, minus 6.3, minus 6.9 kilocalorie per mole respectively. So we are seeing that hypericin was active in terms of inhibition or binding energy. This is very lower compared to the other chemicals. And this is minus 9.1, and the lowest activity shown by thujaplicatic extract, the binding energy was minus 6.3. Stable protein ligand complex formation was due to high binding affinities of the ligands to the diabetes to the phospholipase toxin, and hypericin was found very active against the target protein phospholipase A2. So admit profiling signify the usefulness of the phytochemicals as antidotes against snake envenomation. So finally, thank you. I acknowledge Drug Discovery Webinar 2022 Organizing Committee, Scholars Conference Limited, organized by, for the recognition of my research and supporting me for presentation. Thank you. Stay safe.